So today we are going to go over gene expression regulation and a variety of topics to go over. First, we're going to go over transcription factors. And so transcription factors, they control transcriptional regulation. And so they control RNA polymerases binding to promote and carry out transcription. And there's a couple of general categories. Those are general transcription factors, sigma factors for prokaryotes, and basal transcription factors for eukaryotes, and regulatory transcription factors are gene specific, and they are either positive or negative regulators. So positive transcription factors will enhance transcription when they're bound to the DNA, and negative ones will suppress transcription when they're bound. Sigma 70 is complex protein helps RNA polymerase to bind. It itself binds between -30 and -10 before that so many nucleotides before the promoter it binds, and this is where the initial unbinding will take place, and it's sequence specific, but it's not. That doesn't mean it's an exact sequence like it binds to a specific what's called consensus sequence, which is basically a best match a bunch of sequences very similar. And they can bind to a lot of different gene codes. And because of the variation of the types, it can vary its affinity. So for gene regulation, prokaryotes, operons are a set of genes that are transcribed under the control of a single promoter. And so activators can increase transcription, and repressors can prevent transcription, bind to the operator section of them. And these repressors and activators can turn on. So these operons, the promoter, so there's a couple different places where they'll tripped up. So the repressor the operator is where the DNA repressor will bind to, whereas the activator will bind behind the promoter, like in section laps, or no, behind the promoter. And the there's a couple different types of operons. So there's inducible and repressible. Inducible, the operon is usually what can be turned on in the presence of a specific molecule. Repressible is on by default, but can be turned on in the presence. So trip operon is repressible as well. So I just wanted to put this quote in: "Don't even trip about the triple," because I know that it can a little thing at times, but we'll get through it. So the trip operon, there's a few different scenarios. First, I'll touch upon. I'll tell you. So tryptophan is an amino acid. It's the one of 20 charge you guys get. And bacteria, if they don't have access to it, they need to make the proteins that will produce the tryptophan itself. And so in cases when there's no tryptophan available, like low levels, the genes one to make the tryptophan, but in high levels, this is our co-host. Well, it's working well. So, high levels, if they have access to tryptophan, so here's a diagram, just like an overall picture of the one here. We're going to be focusing mostly on like so the genes in the operon that make tryptophan are downstream. We're going to be looking at this is the promoter, this is the operator, and this is the leader sequence, and this is the place we'll be focusing on. Oh, and so when there's like low levels of tryptophan, there's no tryptophan around to bind the promoter, or sorry, to the repressor, and so it won't bind to the operator to stop transcription. But when there's high levels, tryptophan will activate. It'll bind to the repressor, and then it'll bind. The repressor will bind to the operator, and then the operator, because it's in a way, RNA polymerase can't transcribe it, being genes downstream. So there's a few different scenarios we're going to go over. The first one is high levels of tryptophan, which we kind of already covered, which is basically that when tryptophan is around and there's high enough levels to like bind the repressors, then the repressor will be bound to the, it'll be active and will be bound to the operator over here. And so the RNA polymerase will be blocked from binding to the promoter, so we can initiation. Oh, yeah, so we'll do a little review of attenuation. So this is kind of like the part that's really confusing. So attenuation is the early termination of transcription. And so when there's 4 different regions in the attenuation sequence that we'll be looking at, 1, 2, 3, 4. And 1 and 2 compare together, 2 and 3 compare together, and 3 and 4 compare together. But the outer 2 is like in a single situation, there'll be only 2 situations that will compare. Either the outer 2 pair or the middle pairs up. Oh, yeah, and this only works because these are called hairpins, and prokaryotes can only do this because they transcribe and translate simultaneously. So, like, we'll see down the line that, like, the RNA can be, like, yanked out during translation. It makes a little more sense here. Hello, welcome. You're welcome to stay. Are you sure? Oh my bad. Anyways, the show must go on. Okay. So in low levels of tryptophan, what will happen with the operon? Will transcription occur? Will attenuation occur? Will tryptophan be made? I want to answer that. Yeah, that's right. And will attenuation occur if transcription occurs? Like scenario 2 out of like out of low, medium, and high levels of tryptophan available to the cells. That's like So attenuation does not occur when uh, attenuation does not occur when transcription occurs, because it means that the ribosome and ribosome and RNA and or the RNA polymerase that are downstream of this linear sequence for attenuation. And it so, and why does this occur? So in scenarios of low and medium levels of tryptophan, in high levels of tryptophan, the repressor is going to stop RNA polymerase from which the sequences that do attenuation. But in like situations where there's that's when you're going to choose two scenarios of which hairpins you're going to form. So for low levels of tryptophan, it's going to be expressed, and so attenuation will occur. And how exactly it happens? So in low levels of tryptophan, there is the so the ribosome is going to go down. The RNA polymerase is going to go down, starting from the beginning of the leaders region, and then as it goes down, the ribosome is going to get to a point right before one, and at section 1, there's going to be a codon. Yeah. So right. So transcription's occurring. RNA polymerase goes down. Then RNA polymerase, after doing transcription, it stops after section 2, and there's gonna be a pause sequence there, and then it allows for the ribosome to catch up to its translation, but the ribosome is going to get stuck on this trip codon right there, and when it stops, after it gets there, it's the RNA polymerase that was on holding down 2 is now going to continue on transcribing until it's transcribed the RNA for 2 weeks, and then it's going to stop doing it. It's going to keep going down here, but this ribosome is stuck. Then eventually the this will catch up, and it'll be able to continue with the transcription translation. But so in that case, it's like, but in the case of medium levels of tryptophan, the best thing. Oh, someone already asked. Is a trip operon repressible or inducible? So the trip operon is inducible because it's on by default. Repressible, sorry, because it's on by default, but it can be repressed in the presence of trip. That makes sense. Does that make sense? So what do you guys think will happen if there are medium levels? I'll find out. So in medium levels, initially the tryptophan is so that doesn't occur. Like, the compressor doesn't bind itself to the operator and stopping RNA transcription. So initiation of transcription will occur, which like question on the exam. Yeah, so transcription can start, it can initiate, but then the cell like realizes tryptophan, or there actually isn't around, and so it doesn't need to make, or doesn't need to make form. So attenuation is going to occur. And so the I just made this little neat. You guys enjoyed it. Understanding its so hilarious. So in situation 3 with medium levels of tryptophan, the ribosome is going to, the RNA polymerase is going to keep transcribing, but the problem is that there is going to be, so the RNA polymerase is here, it is able to transcribe down, and then it pauses, or yeah, pauses right here. But then the ribosome is going to be able to quickly move past the trip codon right here at the beginning of 1. And so then it's going to get stuck on this stop codon right here before 2. And then the this allows, and then 3 and 4 are going to get transcribed. And then this allows, while it's on 2 though, it's going to allow 3 and 4 to hairpin. And then it's going to continue on. So before, in the previous situation, it was stopping on, but now it was allowed to tryptophan like transfer. It's like a checking mechanism. And so because it was, then it stopped on region 1. Or sorry, it wasn't stopped on region, it stopped on region 2. It allows for 3 and 4 to transcribe, 3 and 4 hairpin right here. And then because this occurs downstream, you'll see that after this occurs, 1 and 2 will hairpin, and then when it continues to try to transcribe or translate, it'll yank it'll yank the RNA out of the RNA polymerase and transcription transition. Yeah, and so here's like a summary of all 3 situations. So high levels of tryptophan, attenuation is not happening because the repressor is binding the operator. So there's no initiation at all. Situation 2, where there's low levels of tryptophan, the repressor is not going to be bound, and 2 and 3 will form, they'll hairpin, so there's no attenuation. It's good for tryptophan. And there's going to be full translation transcription. But with like medium levels between low and high levels of tryptophan being around, transcription is not for the repressor for the operator. But 3 and 4, section 3 and 4 are going to the hairpin is going to form, so attenuation is going to occur. And this will stop transcription. It's a little confusing, so this might be a little, but I think that there's a little bit of confusion sometimes with what is stopping. And so in like the medium situation, or sorry, let's start with the low. So with the low situation, the ribosome is like pausing or like stalling during the translation at the codon right here. And that allows the going. And then 2 and 3 is going to form. And so as you can see right here, like they're kind of like on the opposites. Like this situation over here at low level is going to lead to this situation because the ribosome is going to be here. And so it's going to not allow 1 and 2 to form because of that 2 and 3 form. And remember, when 2 and 3 form, that is going to allow transcription translation. But in another situation like media level now, RNA polymerase is the one that's doing the stopping, that's preventing this from occurring. So RNA polymerase is stopping at the boundary here and at the beginning of section 2, and this allows for the hairpin of 3 and 4 to form, and attenuation is going to occur, which stops transcription translation. Any questions? Question. And they have many roles on the cell, such as catalyzing actions. So we're gonna go more. Our ribosomal RNA because translation, our mRNA. And then some other ones are small nuclear RNA, which are part of the spliceosomes that do the splicing out of the introns. Long non-coding RNAs are involved with X chromosome activation. CRISPR RNA and do some research for guide strands to insert different DNA sequences. SRP used in protein translocation, which RNA interference is covering, which is post-transcriptional control of mRNA and suppressing which mRNA translated. And so RNA interference is a risk. And so this protein key for finding microRNA. There's 2 different types that are used, mRNA and siRNA. We'll go into those different functions.
Okay, if you don't know. And this is for people in chat too. Right from chat, please. Okay. What does that do? What does that mean? What process is it involved in? Good, yeah. Okay, so essentially, this are all.
Oh, the sound keeps cutting out. Trying to catch my AirPods.
Hello, can you guys hear us now? Yeah.
Okay.
Oh, it was.
Yeah, it's
fine.
probably
Here.
that. My bad.
Okay, cool. No, wait, I wasn't done yet.
Oh, my bad.
I was just, we're just talking. Okay, cool. All right, so just the general mechanism of the RISC is that it unwinds and it binds to the guided strand of the RNA, right? So then once it's binded to them, then that allows the RISC to target the mRNAs using the complementary base pairing. So when you're thinking of this, just think of mRNA. That's important. They're kind of go hand. Okay. And then once it's attached, the mRNA is either going to be degraded or their translation is going to be regulated. So it's kind of what I was talking about earlier. Cool. Yeah, there's two different pathways based on what type the RNA the guide strand is. You guys know what the two pathways are? Anyone? Anyone in the chat? Dave, do you know what the pathways are? Yes. You want to take a guess as to what siRNA does? Yeah. Yeah, so exactly. siRNA is essentially, that one is where like the genetic material is dangerous to the cell, and it's going to be ubiquitinated and marked recognition. What? Any questions? Questions? Yeah. How long is it? Okay, microRNA. What can you tell me about microRNA? Anything. You can read off the slide if you really want to. Just tell me about microRNA. Yeah. Cool. Okay. So yeah, so microRNA is just one of the guide strands, and it's used by RISC. I hate calling RISC. Okay, that was good. Good. Yeah, so it's guided. It's used by RISC to target and base pair with mRNA. So when we're thinking of RISC, remember we're thinking of mRNA, and you can think of microRNA involved in the process as well. And that base pairing just allows the regulation and the stability of the mRNA. But yeah, are these double-stranded or single-stranded? Yeah. No, they're double-stranded. Yeah. I guess that's fair. Okay. And do you think this is used more than siRNA? Which one's used more, this one or siRNA? Yeah. Yeah, siRNA is used more than mRNA. Cool.
And these are to suppress endogenous genes, which means that they're ones that come from within the genome.
Yeah.
So this process starts like in the nucleus. And as you can see here, this diagram shows the pathway.
Okay, so this is the pathway. I'm assuming this would this be provided on the. Okay, so know this. I guess it won't be provided like that. All right, so RNA transcription. Okay. Someone describe the pathway. Joshua, describe the pathway to me. Yeah, just tell me, what are we looking at? Yeah.
Yes.
Cool.
Thanks.
Anyone in the chat? Nothing in the chat, yeah.
No. -20 points for the discussions.
Okay.
Just kidding. Yeah, keep on.
Okay.
Also, just keep in mind this diagram, Laura talked about how in this is supposed to say PRI, like I don't even pronounce it, but PRI mRNA will be cut through Joshua processing, which is shown as the scissors here, and it's going to be cut into pre-miRNA, which is going to exit the nucleus and then will undergo dicer processing.
Well, that's in the second cut. So both. Yes. All right, yeah. So just what Dave was talking about. Once it's out of the cytosol, it's going to go through the dicer, and that's just another RNA nuclease. And then after that happens, they become like these duplexes. And those are small. They're about 20 to 25 base pairs. And then that's what can be bound to this. Right. And then the final step, the guide strand is the complementary base pair for the target mRNA. Yeah, I think it's like that. It doesn't degrade and chop the mRNA because you want, yeah, because there's not always an exact base pair. Yeah, any questions on this slide or on the pathway? Slide. Okay, siRNA. What can you tell me about siRNA? Anything. Okay, tell us something about siRNA.
If I recall right from the lecture video I watched 20 minutes ago, I believe I'm just
Say that.
just kidding. I'm just kidding. That's part of the performance. There's jokes and 20 minutes ago we were still presenting. So siRNA pathway starts in the cytoplasm because it's from exogenous DNA rather than endogenous DNA. So it's not going to come out of the nucleus. And so that's why it started in the cytoplasm. So it's double-stranded or shRNA, which is going to be cleaved by dicer. And then it's going to have this, it's going to be this siRNA duplex is going to be it's going to be unwound and picked up with wrist and ago. Ago and then wrist and it's on there. And then this wrist and siRNA complex forms, and it's going to be base paired. Oh yeah, this complex is going to base pair with mRNA in the cytoplasm. And unlike the miRNA pathway, the major difference is that it's going to be one of them is that it's going to be an exact base pairing match. And it's going to result in the cleavage of the mRNA that it targets by arginine-2, a subunit of RISC. And so it's going to lead to mRNA degradation or silencing. But do keep in mind that one siRNA guide strand can cleave many target mRNAs.
Cool. Wait, don't switch it yet. Okay, before we switch, we're gonna do a fun little quiz. There's only two answers, two possible answers. It's miRNA or siRNA. Okay. Which one is endogenous? miRNA or siRNA? So that means that it starts in the nucleus. And anyone in Zoom, they can answer too. Yeah, so miRNA is endogenous, it starts in the nucleus, and siRNA is exogenous. Yeah, so where's exogenous? So what does that mean? Where does it start? In the cytoplasm. Yeah. And which one has Drosha processing? Yeah. And siRNA doesn't have Drosha. What does it have instead? Wait, what did you say? Starts with a D. Yes, dicer. Cool. And yeah. No, just siRNA. Right? Am I losing my mind? Maybe.
They both have dicer. Dicer processing occurs at both. So right here in the mRNA pathway, it's going to occur later on. It's first going to need an initial processing to go from PRI. Again, this labels here PRI mRNA to pre-mRNA. And then once it's exported from the nucleus, dicer processing will occur when it goes to cytoplasm, whereas siRNA already starts in the cytoplasm. So its first step is going to be cleaved by dicer.
You why would they ask my last question? Okay, pretend you didn't look at the screen. Which one does not need exact base pairing with mRNA? Yeah. Okay, perfect. Yes. You this side? Okay. Mhm.
Okay, so it can bind, it can bind to many, it can bind and cleave many, or cleave many mRNAs of the same nucleotide sequence. Whereas miRNA is going to target, that complex reverse is going to target, it can target a variety of mRNAs with slightly different mRNA sequences. It's kind of like a consensus sequence that we were talking about before, where like, well, yeah, it's because it can go to like many mRNA strands. So like if you have like an mRNA strand that's like AGG, whatever, right? Like with miRNA, it could be like slightly off and still like AUG, whatever, and like still like targeted. Whereas with siRNA, it has to be like an exact like base pairing match. But once it like binds to that mRNA and like leads to its destruction, right? It can like move on to like thank you next, and then like go to another mRNA of the same exact sequence, bind that, cleave it, move on to the next one, thank you next again, and then you see what I'm saying? Does that make sense? It also can. It also can, but it's the difference is that it's not. It doesn't have to be an exact base pair match with the target MO. That's the difference, and the difference is that it doesn't always lead to like direct mRNA cleavage, like in its mechanism, right? In the siRNA pathway, Argonaut is going to cleave mRNA, whereas in the miRNA pathway, the base pairing is going to lead to translational suppression or P-body localization or silencing fragmentation. Does that make sense? Yeah, I mean, like they both like overall lead to like translational suppression. It's just like one of it like directly takes it out, whereas the other one like just like tags it like sense in it. Or it can just like lead to like nothing or lead to nothing translate. The Peabody localization of what they're saying is not like fully understood, and then translational depression suppression is I believe it's just like tagged to that, or it can be like tagged to be like distributed. Okay. Would you have any other questions? Okay. Any questions in the chat? Nothing in the chat. Okay.
Are the socks still on? Did we knock them off yet?
Any more questions? No. Well, thank you for coming. Please enjoy these memes that I found off the web. No, okay, I did not find these off the web. I found this one off the internet, but the rest of them I made.
Did you actually make this one?
Yeah.
Oh, that's pretty good.
Right? Yeah, thank you. I just went to like memegenerator.com. Thanks for coming, guys. Like and subscribe. Oh, does anyone have? I'll stop sharing and stop recording now. Oh, wait, no.